So just to be clear, when doing section 3, you're taking the gene handouts, and for all 3 of the genes, the FTO, RNA polymerase, and the ribosome, for all 3 of these genes, you need to take the DNA and convert it to RNA, just like you see here, and you'll have your RNA codons. However, remember, you're only supposed to take the mutated codons, the ones that are different between Cody and Dylan, and turn them into amino acids. You should not determine the amino acids for each codon, only the mutated ones. And those are the ones that are different between Cole and Dylan. If you notice, this mRNA codon right here, GAU, is different from GAC. So one of them has a mutation. So I solved for the amino acid, ASP. And then we have ASP. But if you look at the next sequence, it's CAG, CAG. They have the same the same DNA sequence, and they'll have the same mRNA as well. It's going to be it's going to be GUC. And again, the same here. GUC. And also don't forget, this is for a specific version. Let me zoom out so you can see this a little better. Keep zooming out. This is for version A. So don't confuse what I'm doing in the video for what you should do for your version. There's version A, version B, as you can see, and version C. Alright, but back to A, right? So for A, we have our notes, right? We were turning DNA into the mRNA. We took the CTA, made it GAU right here and right here. Let's zoom in. All right. But this mRNA codon right here, GUC, is the same as this one right here. If it's the same, there's no need to make an amino acid. There's no need, because there's no difference between their DNA. And that's why you should ask yourself, again, ask yourself, why are you only supposed to convert the mutated RNA codons? Why are you only converting the differences between Dylan and Cody into amino acids? Why? Look at section 2 and connect that to your overall goals. So why would you only worry about the mRNA codons that are different between Dylan and Cody? Why is that? And here's section 2. Here's your hypotheses. So think about this. You're trying to make hypotheses that for different hypotheses that try to find differences amongst the twins. So if you're trying to find differences among the twins, why would you only care about the mutated ones, right? So it's just a simple question of why don't you solve for the amino acids for all? Why are you only going to make amino acids for the mutated DNA? Think about your problems, think about the hypothesis, and then answer this question below. It says here, after translating mRNA into amino acids, and only doing the amino acids that have mutation differences between Colin and Dylan, right? It says you must determine if any of your original hypotheses from section 2 are no longer supportable or if they lack evidence. So what does that mean? Well, in this video, I'm not going to transcribe and translate all the mutant sequences for you. You have the skills to do that, certainly, and we can do that in person if you want help. But if you find that you do all the mutant sequences and you have all the amino acids that are produced from the mutant sections, and they're all the same amino acids on both sides, if you see that every single codon has the same amino acids, then what you need to do is rule out, and you can no longer believe that RNA polymerase is to blame. All right, let me repeat that. So after you've solved for the amino acids for Dylan and Colt, and you find no differences in amino acids, that means that this gene cannot explain that the twins are different. The twins will only be different if you find a gene, whether it's the ribosome, RNA polymerase, or FTO gene, where you find a difference between the amino acids of Dylan and Colt. Any gene, FTO, ribosome, or RNA polymerase, any gene that has the same amino acids top and bottom, you have to rule that hypothesis out, because if it's making the same protein for Dylan and Colt, it can't explain why they're different and look different. It can't explain why their phenotype's different. So what you need to do is, once you've solved for the amino acids, or you've translated the mRNA into amino acids, look at which genes have different amino acids, and there are ones that might have evidence supporting them still. So it says, explain what action have you taken for each hypothesis. It says, explain why you modified or eliminated or kept an older hypothesis. So after you take all the DNA sequences and you determine which gene is showing differences between them, you have to eliminate all the other ones. So if this is RNA polymerase, just for an example, let's say that RNA polymerase said that it was responsible, but we found, let's pretend, I'm not saying it really is, but let's pretend that, let's find the RNA polymerase. All right, let's pretend that the RNA polymerase didn't explain how they're different, that the amino acids are all the same. What I have to do is I have to look at this hypothesis now and say, well, I didn't find any evidence to say that the polymerase was different between the twins. So I can't, it has no evidence to support this. So what I'm going to do is take this RNA polymerase, and I'm going to go down here, and that was my hypothesis 1, and I would just respond. I would say, for the hypothesis of RNA polymerase being different between the brothers, no evidence was shown. Both genes of RNA polymerase for the brothers made the same amino acids. Right, so if it makes the same amino acids, that's basically the protein, and if they're making the same protein, it doesn't explain how the brothers are different. Any other genes, if you find any differences, that means it is supported. So let's pretend that the next gene was, so let's say you found a difference between amino acids. Let's pretend that you found that this was TRY, or tyrosine amino acid, and you found this one was ASP, and that would mean that the RNA polymerase gene does produce different proteins between the two. That should be TYR. Okay. So if that's the case, you could say, oh well, I can't rule this one out yet because there is differences. So another example could be hypothesis number 2 is supported still because there are known mutations and amino acid differences, which is different proteins produced by each brother. These different amino acids may explain their different phenotype, and that is the appearance, right? Observable appearance. There you go. What I'd like you to do is just continue on with each of these hypotheses. So hypothesis 1 through 4, and you may be wondering which one is 1, which one's 2, which one's 3, which one's 4. And I would generally call this one hypothesis 1 and call this one I'll go clockwise. Hypothesis 2. We'll call this one 3. And you can call the last one your creative hypothesis. We can just call that one number 4. So hypothesis 1 may or may not be supported. Remember, what are the reasons for that, right? If it produces different amino acids, you can support that hypothesis. If you have, let's say your fourth one is your creative one. One thing you can still do, let's say that hypothesis 4 is your creative hypothesis. Well, your creative hypothesis, if you haven't, if you haven't found any information to support it, or if you haven't found any information to reject the hypothesis, you could just say, no evidence has yet been shown to support or reject this hypothesis. Therefore, this hypothesis can be kept until evidence supports or rejects the claim it makes. There you go. So any hypotheses you have, if you find out that there's not evidence saying it's not supported, or if there's no evidence supporting it, or there is a lack of evidence to support it or reject it, you can just always maintain it by saying there's no evidence yet been shown to support or reject this hypothesis. Therefore, this hypothesis can be kept until evidence supports or rejects it later. All right. Once you have your hypotheses done, you want to get signature by me. All right. So once you decide which hypothesis here, hypothesis 1, 2, 3, or 4. All right. Once you get my signature on the state of your hypotheses, which you think have evidence to support them and which ones don't, or which ones you're maintaining because they haven't been rejected yet, right? You get my signature, and then you choose the hypothesis you think is most right. So choose, it says for each remaining hypothesis. So rather than doing multiple reports for section 4, just pick the one hypothesis you think is the best. So out of these 4, the one for the RNA polymerase gene causing it, the ribosome gene causing it, or the FTO gene, or the your creative one. My creative one here is Dylan eats more food than exercises less than Cole, right? Whichever one has the most evidence to support that possibility, you choose that one, and then you proceed to trying to complete section 4. Section 4 is essentially a report where you describe different things that you've learned or research you will do to better explain why Dylan and Cole are different. And it's based on whatever hypothesis you're choosing. Right now, you would go right here. And this is the current project page for it. And we have resources. These are links. Links that will take you to a place to help you learn about certain things that you may need to learn about for the FTO gene, learn about RNA polymerase, or the ribosome, so that you can better explain in your report what might have happened between the brothers. It also has links that will help you better understand identical twins, the mutations, and how the twins could have potentially become different. Here is a link to the samples that you've been looking at. That's the same as this page right here. Here's a video, which you've probably already seen, and this video kind of picks up where that one left off. There's 2 different argument forms here. Argument 2 and argument 3 are different. Argument 3, or Google 3, gives you a little more help on which resources up here you should use for different parts. And let me show you what that's like. So using it as a better form, right? So we have 4 different slides here that you're going to use. Each slide is going to be its own little mini report. And it says requirements. You must produce an argument for each remaining hypothesis. Well, we're working on that. Our first report that we'll do is a description of how genetically identical twins could have any nucleotides different from one another. So how was it that identical twins became different in the first place? So this is a slide where we would try to explain how we think the twins became different even though they're identical. Wait for the next slide for more.